Siddharth Damia. The topic for presentation is the future scope of synthetic biology.
Synthetic biology. Synthetic biology is a new area of biological research that combines biology and engineering in order to design and build new biological functions and systems in cells. So how does it work? In synthetic biology, you can think of a cell almost like a computer. In a computer, there are a whole lot of individual parts, like memory, CPUs, and video cards, which work together to provide different functionalities. In cells, there are also a lot of different parts, things called membranes, mitochondria, ribosomes, enzymes, that work together, providing different functions to make the whole cell work. All of these different parts are coded for by a cell's DNA, the same way computer programs have a code to make them work. And in the same way we use different bits of code in different computer programs, we can also use different bits of DNA to program a cell. We call these bits of DNA biobricks. These biobricks can perform a range of tasks such as construction scaffolds for complex biological structures, electrical wiring, and even on-off switches inside the cell. The biobricks can then be connected like computer circuitry, and the synthetic biologist then arranges these individual components to engineer a cellular production line designed to run at maximum efficiency. So what makes synthetic biology different from plain old genetic engineering? Well, in genetic engineering, a few genes might be added or deleted from a cell, but these genes already exist in nature. In synthetic biology, we are making biological parts that don't already exist in nature, either by extensively modifying existing DNA code or by creating entirely new bits of code that produce new components. This is like writing a new program for the cell and getting it to work properly with existing DNA and cell parts. So what might synthetic biology be used for? One example is creating cells that produce special high-tech products that humans can harvest and use. Currently, synthetic biologists are exploring ways to make low-cost drugs that will overcome global shortages for diseases such as malaria. They are also investigating ways to turn sugarcane into jet fuel and create super strong, super light textiles like spider silk. The cells are placed inside vats or fermenters and are kick-started into action, soon to begin producing large quantities of the products they have been engineered to produce. Despite the modifications to a cell's genetic material, the process is safe because the cells are designed not to reproduce outside of these specially controlled artificial environments, making them a safe, environmentally friendly, renewable mode of production. In the future, synthetic biology could be used to alert us to the presence of environmental toxins, produce carbon-neutral fuel sources, and help us diagnose and protect us from disease. Right now, however, there is still a lot to learn because although we know how to read and write the genetic code, we still don't know how to use it to solve a particular problem. But as we learn more about synthetic biology, we will be able to design more complicated cell systems and tackle many of the problems facing our modern world today. In theory, the uses and applications of synthetic biology are only limited by our imagination.
When a new technology is developed or being developed, we want to ensure the safety, so we follow certain rules. Some of the rules are standards, isolation, testing, redundant systems, surveillance, licensing/education. These practices are being used as biological safety and security measures, especially since 2004. The technology is getting exponentially cheaper, so it gets easier to innovate. We need to have better and better tools to track the dissemination of information, chemicals, etc. New organisms are being made genetically, though they are isolated and therefore did not spread. These are organisms that cannot change the genetic material successfully. In the field of computing and biology, we have the trend lines as shown above. It is called Moore's Law. Moore's Law is a trend because the line can deviate at any point. The blue line represents various biological things such as sequencing, synthesis, etc. In electronics, Moore's Law was valid for a certain number of years, so it seemed like a law. Electronics and biology seems very similar. We deviated from the law. It did not take 6 decades to reach down the graph, but 6 years. In 1965 it costed $3 billion for a genome, and now it costs $1,000. This graph represents the time it takes for a new technology to become acceptable in a society. Controversial technologies in synthetic biology and the companies which were set up due to these controversies were Recombinant DNA Moratorium, Genetic, Biogen, NIH Human cDNA Patents, Human Genome Sci, Genome Therapeutics Corp., Human Embryonic Stem Cell Restrictions, Cell Dynamics Incorporated Iparian. This graph shows the modification of human genome through various phases. The techniques used is gene therapy clinical trials. The results for the same are being accepted and implemented in Europe at basic level. It costs $1.6 billion. The experimentation paved the way to cure the diseases through gene modification. One of the example is long-term control of HIV by CCR5 stem cell transplantation. We delete or damage both the copies of CCR5 gene and replace them with manipulated one. Similar progress is being made in field of cancer. This was achieved through targeted gene therapy. Nowadays technology like CRISPR and equipment like programmable nuclease are being made. These are zinc finger nuclease. It was phase 2 research excluding CRISPR. Personal Genome Project was founded in 2005 and is creating public genome health and trade data. Synthetic biology consists of multi-factor dataset. The data submitted by the user is re-identified and examined. The dataset also includes Stem Cell Biobank. Out of this dataset came the Brain Cell Initiative, and it became possible to modify brain and its neurons. This could be done in very short development time frame. The brains can be programmed in C2 neuron technology, or we can build brain outside of the body. Finding of the genome by the nucleus is like finding a needle in the haystack. These nucleases can be targeted not only on humans but also on other life forms. The chart shown shows where these nucleases can be targeted. The diagram shows the kind of achievements achieved till now. The people are becoming more open to the idea of releasing such modified organisms in the environment. The gene drive shown above is CRISPR based. The generation for similar gene drive has been achieved for yeast. This method is directed at sexual transmission, where you can correct and spread a desired gene modification in wild population. It can spread packages such as resistance of the mosquito to malaria. The other possibility is the transmission of only white chromosome is possible, which would result in population depletion of the mosquitoes. It is possible by introducing gene drive chromosome. The diagrams shown show very clearly how this is done using the CRISPR technology. We can conclude that using this technique, all the offspring will have the copy of gene drive chromosomes. The diagrams shown show very clearly how this is done using the CRISPR technology. We can conclude that using this technique, all the offspring will have the copy of gene drive chromosomes. 700 genes together decide the height of the person, but if we see the extreme of the bell curve, we see that the height is determined by a prominent gene. We will be able to fix this using genetic modification in future. If the growth hormone is continued be given to a person even after a certain age will lead to many consequences, but the person will continue to grow until a certain stature. Through genetic opportunities and using rare protective alleles will lead to enhanced muscle growth, decreased body fats and decreased atherosclerosis. These techniques have been implemented on humans and were successful. These techniques don't seem to have downsides. The human lives will be prolonged using genetic modification. The genes of the people who have lived above the age of 110 are being researched. Radiation resistance can be improved 100,000 folds using for genetic mutation. This phenomena cannot be applied to humans right now. Germ-free chickens and goats and conscious insensitive to pain can also be achieved. The above might seem impossible now, but today impossible has become nothing but a word. The things which seemed impossible are being used or researched upon. With the breakthroughs such as CRISPR, everything and anything is possible. Think.